Is it working? Yes. Okay. So let's start again. It's a pleasure to introduce Professor Luisa Gigante Carvalheiro. Luisa is a biologist by training with her bachelor degree from Universidade de Lisboa in Portugal. And she earned her PhD degree in ecology from University of Bristol in the UK. After that, she was a postdoc researcher at University of Pretoria in South Africa and a researcher fellow at the University of Leeds in the UK. And then a young talent fellow at the University of Brasilia, Universidade de Brasília. I try to say the things in the language they are. When I arrive in Portuguese, I do completely wrong. I don't know why. And currently, Luisa is an adjunct professor in the ecology department at Universidade Federal de Goiás. In her lab, they are interested in understanding how environmental changes are affecting biodiversity through time and space, and how such biotic changes affect ecosystem functioning and ecosystem services. And finally, how complex network of ecological interactions in which species are integrated mediate such changes. So please help me to welcome Professor Luisa Carvalheiro.
Thank you. Thank you, Flavia, for the presentation. Hello, everybody. Thank you very much for inviting me for this, for participating in this training program. It is a real pleasure to be here. And so today, as a contribution to this training program, I'm going to tell you a little bit about some quantitative methods that I've been using and that have been contributing to develop my research projects, which focus on what the title of my talk says, so predicting the ecological and socioeconomic consequences of these global changes. So instead of going in depth on these on a specific project, on specific methodology, I opted for a more superficial, to talk more superficially about how I've been using several analytical tools to talk about these, to do research on these four main topics. But of course, if anybody is interested in knowing more about a specific project, feel free to ask afterwards or contact me via email. But the idea is going through these four main topics. So talking about consequences of adding and removing species in ecosystems, then talking about a little bit more how I disentangle effects and interactive effects of global changes, and then looking at how these impacts propagate through socioeconomic networks. And if we have time, talk a little bit about the use of historical databases as a tool for biodiversity dynamics analysis. So when I started, so after I finished my undergraduate course, I had a lot of interest in doing and knowing more about how species interacted with each other and how those interactions help them have success in a given ecosystem. And I was very interested in looking at impacts of invasive exotic species. So I went and did my PhD, and it had a lot of field work. So my early years in the academic, my academic career were really filled of field work. I would go to the field. I would do a lot of observations, mostly on plants and other animals but many groups. I was not focused on a specific group. I was a generalist. I didn't intend to become a specialist on something. I wanted to be a generalist. So lots of field work, but obviously when doing this field work, lots of challenges started to appear because I didn't want to look at the impacts of invasive species on richness or on abundance of native species, on biodiversity. I wanted to look at interactions. I wanted to see how those interactions changed. So to be able to understand that, I started to work with this tool, which is the bipartite networks, which basically use matrix to resume how different consumer species interact with different resource species, and how frequent are those interactions. So we would have these interaction frequencies and then build these bipartite networks, these images where we have resources in the bottom, consumers on top. And these types of tools allow me to then try to play a little bit and simulate situations that could be of interest for management, for people working on management of natural resources. So for example, here, I was studying an area that had these plant species that, despite not looking very particularly attractive, it was a big conservation target for this natural park where I was working, because it was near extinction in the United Kingdom, where I was doing my work. And I wanted to understand how these rare plant species interacted with different flower visitors and indirectly with different plants, and how the exotic species that the park aimed to remove were integrated, were interacting with these plant species via shared flower visitors that were very important for the reproduction of this particular plant species. So using these networks, I was able to simulate what the intentions of the managers of the natural park. So then simulate the removal of the exotic plants in this case, and look how this would impact the flower visitation and the pollination of these rare plant species. So then, as in any simulation, we create a best case scenario and a worst case scenario and intermediate scenarios to then evaluate what could happen. And this was very stimulating for, so this was a part of my PhD, and it was quite stimulating because the managers of the natural park did use these simulations to then adjust their, how they were going to do the program of removal of exotic species and avoid the worst case scenario. But this approach can also be used to guide, for example, other types of management plans, such as restoration. So we know that a lot of Brazilian biomes, and not only in Brazil, are degraded, and therefore there's a recognized need of investing more and more in restoration of natural areas. But most restoration plans, they tend to focus on plants. So the main objective is to restore the vegetation, and they assume that by restoring the vegetation, then the animals will come, so the ecological interactions will also be restored. But that doesn't necessarily always happen. So I'm also got interested in using these tools, these networks of interactions as tools to help planning restoration plans. So for example, in this study here, what we did was, instead of simulating removal, simulating introductions of native species in restoration programs. So we grabbed information of interactions between plants and also flower visitors here. And we then used several algorithms to then try to understand what was the most efficient combination of plant species that would be feasible for managers to use, and that would maximize the restoration of flower visitation, of this important function. So to help these restoration programs become more effective. So this is a more recent example of how I've been using bipartite networks in conservation biology. But when analyzing the impacts of these exotic species. So going back to the topic of the exotic species. So when evaluating the impact of these exotic species on native biodiversity, I started finding some problems. For example, on this study, I was looking at the impacts of the invasion of an Asteraceae plant that is a big problem on the east coast of Australia, where this study was done. And I wanted to see if this plant was affecting negatively the native diversity of not only of plants, but also of the seeds of the fruit herbivores that feed on her, on these native species. So I wanted to look at this, and so this is an example. We had several plots in this study, but this is an example. And just by looking at this plot, so in gray, I have here the native species. In black, I have the exotic. And just by looking at these, we can see that the more of these exotic plant species, which is called birdbush, the less we have, the less native species we have of native herbivores. So if I did a simple regression, it would be this impact would be this negative relationship would be very significant. Yet there was a problem that associated with these invasive plant species, there was also an exotic fruit herbivore, which was this little guy here, which is called Mesoclanis polanis, which was an introduced biocontrol agent that was not very successful controlling the invasion of the plants of birdbush, and instead also became hyperabundant in these native ecosystems in Australia. So instead of having one exotic species, we now had two exotic species, one that was intentionally introduced. The managers claimed that there wouldn't be negative impacts of the introduction of this biocontrol agent because it was very specialized in their feeding habits. So it only ate this plant birdbush and didn't eat anything else. And indeed, all the flies that we detected of this species, they did come from these exotic plant species. Yet, by using these networks, we identified that there were several natural enemies that were feeding on this exotic fruit fly. And these natural enemies that were parasitoids and predators, they also fed on native fruit flies. So we had shared natural enemies between our biocontrol agent and the native flies. And this leads to potential negative impacts via apparent competition, so via increasing the abundance of natural enemies, which then declines the abundance of the native herbivores. So basically, we now had two different paths that were possible to explain that very clear negative relationship between the invasive plant and the native herbivores. It would be possible that the invasive plant species was reducing the native plants and therefore was reducing the native herbivores. But another possibility was that the invasive plant was increasing exotic herbivores, as we knew it did, and then increasing the native natural enemies and therefore having negative impacts on native herbivores. And it is, while both paths are likely to occur, it would be very important to identify which one was causing more damage in order to define better management plans. So that's what we can do. So we can do that by comparing several different models that try to explain this relationship. So instead of having one single model, like a simple regression, which is the most simple linear model that we can have, we can compare multiple models, and we can even do a path analysis to understand which of these paths is more likely to explain the negative impacts that we were detecting on native herbivores. So that's what I did in this study. So I compared different models. I did a model selection to identify which path was having most important impacts. In this case, we detected that the path that was having the most impact was via shared natural enemies. So it was because of the introduced biocontrol agent and not directly because of the invasive plant. So this was an important result. And it's also an example on how general linear models can be important to help us understand which paths are more likely to occur. And also, this tool, general linear models, can help build predictive models that can then be used in future to predict impacts. But invasions are not the only threat to biodiversity. We have many potential predictors. So it's not only invasive plants or any management action that is done towards these invasive plants, but also many other predictors such as land use changes, so the loss of natural habitat. We have different types of climate changes that are occurring. We have important changes on biogeochemical flows. So for example, the levels of nitrogen, phosphorus, and other nutrients are changing substantially because of human activity. And all of these have the potential to affect biodiversity, and they may also have interactive effects between each other. So when two drivers are affecting biodiversity, the effect of them together might be more than just the sum of the effects of them separately. There might be synergistic effects that amplify the effects of a specific driver. So I become more and more interested by these multiple drivers. And so after I finished my PhD, I decided to change a little bit my approach on the study of biodiversity. And instead of focusing on one driver, so I was studying biological invasions. So instead of focusing on one driver and look at multiple groups of species, I decided to focus on multiple drivers, but then I had to focus on a specific group of species. And with that approach, I want to then talk about these interactive effects of global changes. But before, I needed to choose a group to focus. And I decided to focus on pollinators, and I want to tell you a little bit why before the types of studies that I've done. So I'm very interested in all types of interactions, ecological interactions, but I have focused indeed my research mostly on pollinators. And the reason why this happened, it's, well, because they are very important for ecosystem functioning, because they are responsible for the reproduction of the vast majority of plants. So if the reproduction of plants depends on them, then any other interaction will also be influenced by these pollinators. But another reason which kept me on working on pollinators, it's because they make a very interesting link with aspects of sociology and of economy. So they have a very important effect on agriculture production. In fact, the vast majority of the products that are cultivated worldwide, they depend in some extent on pollinators. So this has been increasingly recognized, and therefore pollination is nowadays recognized as an important agriculture input, together with water, with the nutrients that farmers need to add, the pest management. Farmers nowadays also think about pollination. And each particular plant species will have specific pollination requirements and will depend on different types of pollinators. And this means that if this diversity of pollinators has an impact on agriculture production, it also has an important impact on food security as a whole and on human health. So basically, pollinators help to make a link between the ecological networks, or the studies in ecology in general, with socioeconomic networks. And I find this link very interesting because this is not a one-direction path. So the pollinators benefit agriculture production, but as we know, agriculture production also negatively affects pollinators. So it's actually one of the main drivers of loss of natural habitat. So for example, to have common food reach to our plates, food that we enjoy every day, millions of hectares need to be devastated in order to produce this plant species on which we depend to have the food that we like to eat every day. Even for meat, so to feed cattle, we need to plant large extensions of many crops. Plus, there are all the negative impacts associated with intensive use of pesticides, intensive use of fertilizers, irrigation, that also causes negative impacts on biodiversity. So basically, pollinators can help by recognizing the importance of pollinators for agriculture, we can then build socioeconomic tools that can help promote sustainable development. And there are several types of tools, either at government level, at producer level, or even at consumer level, that we can think about. And this was something that really caught my interest. And this is the main reason why in the past, 10 years, I've been mostly focusing on pollinators. I still study other groups. I do like them all, but I focus a lot on pollinators. So, as any other group that I would decide to focus, there are many drivers affecting pollinators, and so many potential predictors of the biodiversity or the abundance of this group. And to understand how these different drivers are affecting pollinators, and if there are interactive effects between them, then again, these general linear models can be a very important tool. And they can help us study impacts that are linear, but also the ones that are not so linear. So the general linear models, or the generalized linear models, can also help us study relationships that are not linear. And they can also help us understand if the impact of one specific variable, for example, pesticides, if it depends on the effect of another variable, for example, climate. So it can help us look at these interactive effects. So as an example, in this study, we were trying to evaluate the impacts of air pollution, nitrogen, nitrates in general, and also tropospheric ozone on pollinators and pollination. And there are several ways for which this ozone can affect pollinators. And also there are several potential interactive effects with other drivers, such as nutrient fertilization, climate, pesticide applications, several potential interactive paths. And in this study, with the use of these general linear models, we were able to detect that, for example, the negative effect of pesticide that we expected to find, obviously, was far more accentuated when in regions where the levels of stratospheric ozone were higher. So the levels of air pollution were higher. And this was a very interesting result. We were expecting other types of interactive effects, and we ended up finding with ozone. And this was quite interesting to see these relations between air pollution and farmers' management practices. Another example, here the objective was to look at several benefits that are mediated by biodiversity on crop production. And we wanted to see what was more important, if it was richness or simple abundance. So for example, if we think about pollination, the farmers that recognize the importance of pollination, they may decide, okay, I know that pollination is important, so I want to, I'm gonna rent hundreds and hundreds of beehives of one single species, place them in my plantation, and my problem is solved. And what this study found here is that this approach is not so good because, yes, abundance is important, but richness is far more important. So while the two are correlated, richness is far more important for pollination than abundance alone. And the same when we were looking at pest control. So this is an example on how general linear models and particularly path analysis can be used to identify the most effective paths. Another example here, in this case, we were evaluating the positive effect of pollinators on the productivity of common bean that is so popular on Brazilian diet and the diet of many other countries. And okay, we were expecting this positive effect on productivity, but what we found was that this positive effect was not so accentuated and even tended to become negative when the farmers were using fertilizers in excess, so far above what is needed. And this is a common practice of farmers. They tend to use more fertilizers than what is really needed. And with this study, we were able to show that they lose the positive effects of pollinators with such practices.
Luisa, we have a question. Can they make during your presentation, or you prefer in the end?
No, actually, I only didn't put that option because I thought I would have to do it in a row. But yes, it's even better if we interact during the talk.
You can go, Laimara. Thank you.
Hi, Lisa. Good afternoon.
Hi.
Can you go back one slide?
Yes, I can.
Are you listening her?
I think it failed a little bit, but she asked me to go back.
Is it better now?
Yeah.
All right.
It's better.
What are these numbers on these arrows? Because you said abundance has a higher effect on pollination, but I see the number in abundance is smaller. So if you could explain the graph a little bit.
Okay. So when you do path analysis, for example, you can understand how much of the variability on your on your response variable, which is in this case is pollination, how much of the variability in pollination is explained, in this case, by abundance of pollinators or by richness of pollinators. So it separates how much you can explain. And you can also calculate the effect of each of these variables. So in this case, the effect is stronger. You can put here different metrics. I would have to check here to tell you exactly what this is. But here it's a comparative measure of the effect of richness on pollination. So in this case, the effect of richness is higher than the effect of the abundance on pollination. So I was comparing these two values. Does it make sense?
Yes, but the effect of richness on abundance seems to be pretty big.
Yes. So these two variables are extremely correlated. So the abundance and diversity of in a community are always very correlated, and this was expected. But what we wanted to disentangle is the independent effects of richness and abundance. So we wanted to compare the effects on, in this case, crop production, crop pollination, and therefore production. So we were not so interested on this, but more interested in comparing these two effects. So this was, okay, let me give you a bit more of information. So the data sets behind these are not controlled experiments. They are data that was collected in the field. So we have locations where there is higher diversity of pollinators, and others lower diversity of pollinators, and there's always an abundance associated. And there are regions where you have big abundance and low richness. But because this is natural communities, there is a positive relation between richness and abundance. This was not, they were not orthogonal, they were not completely separated. So this high correlation was expected. But what we want is to see which of these two variables was more important. Does it make sense?
She says it makes sense.
Okay. Yeah, but if you were looking at here, so indeed in here, the effect of abundance and richness is very similar and even slightly higher for pest control, if that was what you were looking. Here it is richness that is higher. In pest control, it's not it's not richness that is higher. Okay. I'll continue. Let me, oops. Yeah. Okay. So this was just another example, in this case, of the effect of how fertilization affected this beneficial effect of pollinators. But this is a local study, as other examples that I gave before. This is a local study. And sometimes if we want to make really recommendations, we need to understand if we can really generalize these effects. So if we can, if these patterns are really global, or if it's something very specific. So for example, this study was done in Cerrado near Brasilia with common bean. Okay, but can we generalize to all the crop species in Cerrado, or can we generalize to all the crop species worldwide? So another type of studies that I am involved is in this meta-analysis or synthesis work that really try to combine information on similar studies from many parts of the world, and then try to see if there's general recommendations that can be made for agriculture, in this case, in order to protect biodiversity. So to do that, we have challenges because, for example, if we are looking in a study that is done in the tropics, biodiversity is naturally higher than studies in for example, regions more close to the poles that are colder. So we have these problems. We also have the strength of the effect may also depend on the specific crop. So there are many reasons of variability that are problematic if we try to combine all these data sets together. So we need to control for this variability between studies that is caused by other reasons other than, for example, pollinators, which was what we were studying here. And we can control this variability in using general linear models by, for example, introducing random intercepts or random slopes. This is another type of variable that can be introduced in this model and allows us to control for this variability between studies, and also allows us to extract a global effect. So we can compare the effects of different studies doing general linear models, either by including random intercepts or by using weighted general linear models. So for example, sometimes we don't have the data, but we have the information on the effect. So we know, let's say, for example, that in a given place, pollinators double the production of one crop, while in another region, pollinators have a positive effect, but it's not so accentuated, and in others even has a negative effect. So there is a big variability, and while we don't have the raw data in order to use this approach, we do have information on the effect and on the error associated with this effect. So if that's the case, we can still compare all these studies using a weighted general linear model, which is normally known as meta-analysis, in order to extract if the global effect is null, positive, or negative. So this is what we did on this specific study. So in this case, we were looking, so we collected many studies that looked at the effects of pollinators, of diversity of pollinators on crop production. And we had studies from all over the world. And these studies involved many different crops from different plant families. The techniques that were used to assess these benefits of biodiversity on crop production were sometimes different. And we gathered all this information and compared the effects and to extract a global pattern. On this particular study, one of the most striking findings was that the effect of wild native pollinators was much more consistent than the effect of honeybees that are usually managed and introduced in many of these areas. Also in the study that I was talking about, that used the pass analysis, this was indeed also a synthesis work that gathered data. In this case, it was the original data from many studies all over the world, and use this meta-analysis approach, but combining it with pass analysis in order to extract a global pattern on the effects of richness and abundance of pollinators and agents of biocontrol. So to resume a little bit of how I use these general linear models, I find this tool very powerful. So the simplest general linear model would be a regression, would be an ANOVA, but you can do much more with this tool. They are very important. They are very good in resuming and controlling variability and determining the best predictors, measuring effects. They can also be used to determine ecological niche. So this is one of the potential algorithms that can be used when you're doing species distribution models or niche models. There are many other algorithms that are also very useful, but GLM are one of them. And they can also be used to measure and predict temporal dynamics of populations or communities. But overall, this is a tool that can help define these global patterns, for example, with the meta-analysis approach, and define predictive models and the error associated to these predictive models. So it can be used to then build stochastic models that can predict more complex patterns, as the example that I'm going to give now. So for example, one of the things that we do with these estimations that we predict the effect of several different drivers on specific response variables. We're using these general linear models, and then we are able to integrate them in more complex models in order to predict for example, in this case, we are trying to predict, this is part of the PhD thesis of a student of mine, Felipe Silva, and we are able to predict the profit. He's an economist, so he's far more interested in the economy side of the question. In this case, he was trying to predict profit that farmers have in investing in sustainable practices of agriculture, or investing in conventional practices such as the use of fertilizers. So we can use this information to build predictive, in this case, socioeconomic models. Which then, this is the aim here, is to then create a tool that can be used by farmers. So this is an example on how pollinators and the tools that I've been mentioning can help promote sustainable development. And in this case, it's targeting the producers. But one of the topics that also catches a lot my attention is to go a little bit further and see how these impacts really propagate through these socioeconomic networks. So as I mentioned before, one of the reasons that caught my attention to study more and more pollinators is how easy it is to make the link between conservation of biodiversity and the ecological networks and these socioeconomic networks. And I've been particularly interested on how the management choices affect farmers, but also I was interested in knowing how they propagate through these economic networks. And to be able to do this, again, together with Felipe, we created this concept of virtual biotic pollination flow, which basically resumes how much of the importations and exportations result from this important ecosystem function and important ecosystem service, which is the biotic pollination. So basically, we wanted to see how much of the work of the pollinators of a given country ends up being consumed in another country. So the basic idea behind this is that any bee or any pollinator can make a specific number of flower visitation events during their lifetime. And part of them go to natural ecosystems, but a big part of them, they go for crops. And part of that product that is produced in those crop fields goes to national park market, and a big part also goes to international market. This product does not totally result on the action of pollinators, because many plants are able to self-pollinate, and also pollinators cannot reach everywhere. So first, we had to use this spatially explicit analysis in order to understand how much of the crop fields is accessible to pollinators. So then we use data. This was a project working with all countries in the world. So we use this SAA land use information in order to understand how much of the cropland was not accessible to pollination, and how much was accessible to pollination because it was sufficiently close to natural areas where these pollinators nest. And based on that, we created this level index of isolation of cropland. And then we were able to calculate this virtual biotic, integrating this with information on exportations and importations. We were able to calculate this virtual biotic pollination flow. So basically, we were integrating information from trade markets with information from the effect of pollinators on different plants. And with that, we then created this tool, this online tool that makes things easier for people to see how much of the work of pollinators goes to other places. So for example, it helps us understand where does pollinators that produce cocoa come from, for example, to produce the chocolate in Belgium and Switzerland. And this can help us think of what would happen. Well, there is a mistake here, but what would happen to chocolate if we lose the biodiversity and specifically the pollinators of certain specific countries? Or we can try to understand where the beans that result from the work of Brazilian pollinators or pollinators of a specific biome, such as Cerrado, where does that work go? The past was to produce beans. We can do that with mangoes. We can do that with all the products that are produced, and then have an overall pattern on how where the effort of these pollinators of Brazil is going to. So basically, it helps us integrate information. It helps us then make the future links on how ecological networks integrate with these social economic networks. And we can then further explore this, as we did in this publication, where we then use again the general linear models to try to understand, to relate this information with information on development level of the countries. And with that, we were able to understand that less developed countries contribute much more to this virtual pollination flow, and the richest countries are the ones that consume more. And we were also able to detect that the countries that are contributing most, which, as I said, were the least developed, but lower indexes of development, are the ones that are replacing more natural habitat by crop fields in the recent years. So this raises questions that can be quite important to think about socioeconomic tools at governmental level. So as I said, I was a bit superficial on everything, but I told you a little bit how I integrate a diversity of analytic tools in order to evaluate and predict these ecological and socioeconomic consequences of global changes. I'm open to questions. But to finalize what I wanted to tell you in this talk, I would like to talk about another topic that has caught my attention quite a lot in the past, especially in the past 10 years. So when studying these global changes, we have a lot of very good information on land use, on climate, on many other potential drivers from many years back, from many decades before. For example, here we are seeing land use maps from the 1985 and 2020. But in reality, we don't have, for many regions of the world, and certainly the Amazon and Atlantic forest, especially the Amazon, is definitely one of these examples. We frequently don't have enough information to know how many species of any group there were in the past. And while with the, for example, land use, we are able to use old maps in order to build these historical maps on land use, with biodiversity, it's very hard to go back in time. We cannot go back in time in order to sample again these regions. So many areas were never sampled during those times, and therefore we don't have a reference to then evaluate how much we have lost. And for many reasons, even for recent years, we still have this problem. So in Brazil, it's a good example, and there are many other examples, and countries that can be good examples, that vast areas are still very understudied, and we don't know how many species we have. So where can we get this data from the past? From collections. Museums collections can help us travel to the past, where there are millions and millions of objects in museums and in private collections. Sometimes they are not yet digitized, but there are efforts of digitization of this information to make it accessible. Also, there are millions of records that have been recorded haphazardly throughout the world, and they may not have a specimen, so they are not in collections, but the information was collected, and we have millions of records in platforms such as GBIF and has Species Link. And each of these records or specimens usually has information on who collected, where it was collected, and when, at least the year, this information will be available. So we have a lot of very valuable information that is frequently stored in drawers, but more and more becoming available via digitalization. So this is the best way we have to travel to the past and be able to estimate biodiversity in the past. However, we have important, substantial analytical problems when wanting to use this historical data. The first problem that we faced is that we have very unequal sampling efforts in the past and in the present. So typically, whatever group you decide to study, you will have much less information from the past and much more information from the present. So this brings me to the last part of my talk, where I would like to talk a little bit how I've been dealing with these problems. There are other problems with historical databases, but how I've been using bootstrap methods, so resampling method in general, species accumulation curves, in order to be able to extract patterns of biodiversity dynamics with historical databases. So how can we extract, if we have this huge difference in number of records, it becomes very hard to compare the numbers of species. For example, if in the past we found similar numbers of species to the present, but we have much less records in the past, then we are not able to say that there is no change between these two points. So what we need is to estimate how many species we would have in the present if we had a similar sampling effort, or how many species we would have in the past if we had a more intensive sampling effort. And species accumulation curves are a very important tool to help us reach this conclusion, to help us estimate these numbers. So the idea of a species accumulation curves, it's if we sampled randomly these communities, the data that we would have collected, the more we sample, the more, as the number of records increases, the number of species that we detect also increases, and this ends up building a curve that ends up reaching a maximum number of species. If we sampled the whole community intensively, then we would certainly reach a maximum number. If we haven't sampled enough, then we would still be in this first part of this accumulation curve where our number of species is still rising steeply. So we can use these species accumulation curves to predict how many species we would have detected if we had the same sampling effort in the present and in the past, by resampling the data that we have and building these accumulation curves, and then making comparisons in exactly the same sampling effort value. So for example, if we did the comparison here, we would conclude that in the present, if we had sampled only 3,200 records, we would have a much lower level of a number of species, and therefore we can estimate that we have a 10% decline in biodiversity from the past and from the present in this example. So species accumulation curves are based on these probability methods, on resampling, and there are formulas that have been developed, that have been integrated in our package. For example, INEX, when I first started using these types, I had to write my own codes, my own R codes in order to integrate the formulas. Nowadays, there's iPackages available that are able to, can be used freely and help us to then predict how many species we would find with a given sampling effort. Yet, these methods have been developed for sampling that was done randomly, so that has not any bias. So it assumes that when a given person goes to the field and samples, it does not have any kind of bias. It just randomly picks what it is there. And when we are working with historical collections, things are not really like that. So typically, the data was not collected in a standardized way. There are far more records near roads, near cities than near natural areas. The way people collect it has a lot to do with their interests, and many collectors are interested in rare species and also want to have in their collection the variation in morphotypes represented. So, for example, the collector tends to have males and females of the same species. It tries to have that. So there is an added effort. There is a bias towards rare species. Then there is a bias towards rare species, and this raises problems when using these species accumulation curves, which assume that data were sampled.
We will wait for her to be online again.
Maybe it's us because earlier today it was us.
Was us? A Natália, vê se está conectado no cabo. No cabo?
Mas terça internet talvez?
Eu também acho que é a dela. Oops.
In the EFT, maybe the internet there is not so good today.
Yes.
Okay. I was worried that the problem was here with my connection.
Luisa, can you hear us again?
Hello?
No, I'm not. Hello, hello?
Can you hear me?
Yes. Can you hear us?
Yes.
It's our internet connection that failed, but you were in this slide when you were talking and you got frozen for us.
And did you saw this slide already?
We saw this slide and then you were in the next one.
I went back to this
Yes.
one. Okay.
She was there, right, guys? Yes.
Okay, so what I was saying is that these species accumulation methods, these rarefaction methods are very useful and can help us deal with some problems, but not all. So I was saying that, for example, these methods depend a lot on the number of species that is recorded once and the number of species that is recorded twice. And because there is a bias towards rare species, these species, most species tend to not be singletons, to have to be overrepresented. So to deal with this, we had to develop a set of rules and ways to deal with this database, with this historical database, to avoid duplicates, to avoid overinflated inputs of data. And also we integrated bootstrap methods that basically resample the data in order to get more adequate estimates of these numbers of singletons and doubletons that are so important in the formulas of these species accumulation curves. and this is something that that was developed. So based on the formulas of Anne Shao, which was one of the people that created the R package that I was talking about, we then developed further functions, R functions, in order to adapt this type of methods to historical collections. And with that, just as a final example to finalize here, we were then able to compare levels of richness in the 30s, in the 50s, and more recently now in the recent decade.
So bad. Let me. Aqui não está conectado e eu estou na EduRAM, na IFT, desculpa. So she's talking. Sorry.
Hi.
Sorry, Luisa. I don't know what is happening with the with the internet today. I can tell that you were in this slide telling about the results that you got from the past and from the present. And
Yes.
I don't know where you stopped after.
Okay, I was just saying that also it's quite important to repeat this analysis at different spatial scales. So this is an example of a study that we did in Netherlands, although we do have ongoing work for Brazil. But this is an example from Netherlands, where we run this analysis at 10 by 10 kilometer grid cells. Then we repeat it. I think this is 80 by 80 kilometers or 160 by 160 kilometers. And then we did the same for country level. And we found that declines when we run at smaller spatial scales that we did not find at coarser spatial scales, which basically indicates that many species are retracting their range. They are still present in the country, but they are retracting their range. So there's quite interesting information on, besides just saying species are declining, species are not declining, there's also information on the dynamics of, spatial dynamics of species that can be extracted from this type of analysis. And obviously this can then be integrated with land use information, with climate data, as we have done, again, for the Netherlands and England as well. And now we are aiming to do this in Brazil. So basically, main message about these specific tools is that they can be, there's still a lot to develop, there's still challenges to deal, but they are ways of that allows us to take more advantage of this historical data, of this collection data that exists throughout the world. And we need to give more value definitely to the museum collections in order to be able to travel to the past using these tools. And that's it. That's the summary of what I wanted to tell to you. So again, apologies for being superficial, but I thought this potentially could be more interesting. Thank you very much for listening, and I'm open to any questions that you might have. I see a lot of hands. I cannot hear anything, but I see a lot of hands.
We were clapping a lot. There are questions, and just to explain to you, people need to pass the microphone for you to listen, so it takes a little bit. Yes, go ahead.
Hi, Luisa. Thank you for the amazing talk. I agree that pollinators are extremely important for agriculture production as well as ecosystem functioning. And the first question I have is, are those arrows in the directed graphs isolated effects, or are they compounded? I mean that richness seems to increase abundance, and abundance seems to affect both pollination and pest control, right?
Yes, exactly. In that past analysis study, yes.
So when taking that into account, I was thinking that richness seems to be enormously important as it increases abundance as well. But we know that monocultures are one of the biggest culprits in reduced biodiversity, especially for pollinators, right? And I was wondering, how can we recommend for producers to invest in diversity of pollinators when monocultures themselves are a big part of the problem?
So I think so recognizing this, so just first your first part of the question. So the two are important for sure, abundance and diversity. So if we have one individual of multiple species, it won't help a lot. And what several studies have found is that sometimes these rare species, they change the behavior of abundant species and make these abundant species more efficient. So the abundant species are still important. How can we convince farmers when monocultures are the big part of the problem? So I think that there are the term agroecology is sometimes used in different ways in different parts of the world, but thinking of agroecology as implementation of ecological principles in agriculture, there are several ways that these practices can be implemented in small scale farmers, as well as in these large monocultures. So the fact that it is a monoculture does not mean, okay, this is irrelevant for this type of production. There are several ways and there are several scientific evidences that show that specific practices can be used to allow pollinators and biocontrol agents to travel further into these crop fields. So in this can improve the productivity of these systems and therefore avoid further intensification in maintaining the productivity level. So there are several suggestions that have been made, for example, flower strips that can be cut a little bit, this monoculture, maintaining the large areas that these large producers want, but cutting them slightly in order to allow the pollinators to come. There's obviously other strategies that imply mixing different types of crops and thus making them less intensive. But even if they kept these intensive levels in terms of size, by reducing fertilizer application, by reducing pesticide application and implementing these flower strips, this would already make these agroecosystems much more sustainable and friendly for pollinators, and may even increase their production. Did I answer your question?
Yeah, thank you very much.
Yes, there's one more question over there.
It's a follow-up on the last question, because monocultures, the really delicate thing about monocultures is that we nowadays have a system of agrotoxics, pesticides. Yeah, pesticides that are needed to maintain the production of those crops, and crops that are genetically modified to resist to some specific pesticides. And so in this feedback, we have some species that become resistant to those pesticides. A higher amount of pesticides are needed, and those pesticides have an effect on the pollinator biodiversity, the biodiversity of other plants that are important for certain pollinators to keep diversity high. And so looking at that whole frame of things, that monoculture is associated nowadays necessarily to the pesticide industry. Is it really possible to enhance the amount of biodiversity of pollinators in those fields, in monoculture, in this system? Is it a real possibility given all those conditions? That's my question.
Mhm. So this is a challenge, and I'm an optimist by nature. So I say, yes, it is possible. We need to think that it's possible in order to continue to stimulate this type of changes. And there are challenges. Obviously, if you have a single plant species in a large field, this will be fantastic for anything that feeds on that. So pests will become hyperabundant. And we cannot control 100% the tests with biocontrol agents. It's very hard. So this is a challenge. And sometimes it is interesting to think in more integrated pest management actions that potentially use pesticides in certain situations, but keep considering as first choice other types of controls. And also, most important, the farmers need to recognize, and this is something that is already occurring, that that there is this other important input other than pest control, other than nutrients, other than water, that is the pollinators. And this is really important for more than 70% of the crops. So really, it is a large number. And what happens is that production, there is this Lidbeck rule. So basically, if you have several inputs that you need, if you are below what is needed for one of them, it doesn't matter that you increase the others more. Your production will still be limited by the lowest input. So if you increase pesticide, you are reducing the level of input on pollinators, and they are adding pesticides, and they are not getting benefits. If the farmers don't know why, they may not stop using pesticides or fertilizers because they don't know why. So one of the main challenges is to make the connection between academy, and I think this is a main challenge for anybody that is starting their academic path, and then it's important that everybody considers is that we need to make a better connection between academy and society. So we shouldn't close in a box and say, okay, I'm an ecologist, I work on conservation, I don't want to talk with farmers, I don't want to talk with politicians, I just want to focus on conserving. That doesn't work. We really need to make the connection. And a way to make this connection specifically with farmers, it's to talk in the language that they understand. What is the main objective of a farmer? It's to improve their profit. For small-scale farmers, sometimes it's subsistency. For large-scale farmers, it's profit, is how much profit they gain. So we need to integrate this information that comes from ecology with information from other important inputs and, in this case, economy, market prices, in order for them to see how much their profit will change if they invest on a particular insume, for example, natural capital, instead of investing in other inputs such as fertilizers or pesticides. In addition to that, we have the potential of developing governmental subsidies that would integrate this framework and increase the profit for farmers if they, in addition to the benefits that they get from keeping the pollinators, they even get a subsidy for protecting biodiversity. So this is the only way to really change quickly behaviors in these intensive farming systems. It's to talk in their language. So I hope I have answered, at least with my opinion, on how to change these systems.
Is that okay, Lemara?
I agree partially, but I will push you a little further. We know, and agroindustry knows about soil loss for decades now. And soil is, of course, water is important, pollinators are important, but soil is the most important thing for agriculture because you can't really renew it basically. It's very hard to do that, and we've been losing soil effectively for decades, and agroindustry knows about that. And nothing has changed, and we've been losing soil for many reasons. We know the reasons, agroindustry knows the reasons. Things haven't really changed. I believe information maybe isn't going to change a lot. I know it's not our work as scientists, but I think it's important to recognize some limits and some... Well, in the beginning of the presentation, I remember you used the words need, like we need to lose soil, or we need to use pesticides, or we need to use a lot of water to produce. And that is not the only way to do things. And we know that. I'm sure you know that. But these little words, I think, are important. I don't think we should say that the production way needs to be that way. It is that way now. It doesn't need to be that way. And I don't see how these changes are going to happen unless they are enforced. For instance, the loss of biodiversity. We are still, at least in Brazil, subsidizing pesticides, subsidizing other ways of production that aren't healthy for our environment in the long term and our society in the long term. So maybe I wish I have seen a little bit more focus on that also, on these changes that need to be enforced, because we've been informing industry for before I was born, and not much has changed. So that was what I wanted to comment on. Yeah.
Okay, so I'll try to be brief on my reply. But first, what I've shown is that there are alternatives, and that's the big objective of the work that I've been presenting, is exactly there are alternatives to intensive agriculture. So you misinterpret my message in there. But specifically on what you said, that there was no changes, I'll now have to reveal how old I am. But I started working on this topic, specifically on agriculture after my PhD. So that was more than 15 years ago. And working with farmers, talking with farmers. And at that time, farmers would, they might be nice to me and give me coffee and everything, but they would completely ignore absolutely that anything that I said about biodiversity or pollinators. There was absolutely no recognition of the role of biodiversity for crop production. And I've talked with a lot of farmers during I started getting interest, and that was the reaction. So I started studying in their lands, and clearly there was no opening for changing any of their practices becoming more sustainable, because there was no recognition of the importance of biodiversity as a whole, or natural resources more generally. Nowadays, it's completely different. It changed a lot. It may not have changed as fast as we need in order to really be able to halt biodiversity loss, but it changed a lot. If I talk to any farmer, if I go, for example, I work with how do you say that in English? reforma agrária, assentamentos. You are Brazilian, so you will know what I mean. I work with farmers from these very small scale farmers. I work with agricultura familiar, so family scale farming, and with the large scale farmers, all these types of farmers. In the past years, I come to any of these farmers and everybody knows that pollinators are important. I get surprised with how much they already know about this. And people even, includes people that have done university degrees and people that have not finished the first years of education. Still, they know that pollinators are important because information circulates. So it changed a lot. With soil that you were talking about, before, people just would do tillage in the soil and not care nothing about soil preservation. Nowadays, the vast majority of systems, large-scale systems in Brazil, they no longer do tillage. They do they maintain the crops they in order to support soil. So there's a no tillage approach to farming overall, even in large scale like soybeans, common beans farms. They no longer do this tillage that was horrible to soil and led to huge soil losses. Is it changing in the speed that we need to stop biodiversity? No. We are doing everything very, very slow, but it is changing, and it changed a lot in these past 15 years that I've been working with farmers. That I can tell you. So we need to continue bringing information from academy to the farmers, from academy to the politics. We need to continue working with that. And if you speak with the vast majority of academics, and I'm included, so in that, I think we don't do that effort so much because we don't have the formation to talk with the non-academics. We are educated in order to talk and communicate with academics. So we don't have the necessary skills to communicate with non-academics. But we should make more of that effort in order for the value of science to be more recognized.
Is that okay? We have many questions, Leimara, yeah?
I was just gonna say thanks.
Okay, thank you, Leimara. We have a question from Zoom, and then I will call you later. Gabriel Marins, can you make your question? Or you...
Hi, Luisa. Good afternoon.
Hi.
I'm Gabriel. First, I wanted to thank you for your presentation. It was really good, really inspiring. I work with an ecological network and landscape ecology approach to the relationship between biodiversity and pest control. So a little bit of agroecology. And after this presentation, my mind's kind of blown of the tools I can use. You mentioned a lot of them. My first and direct question is, can I reach to you to know more about how to use them and what I can answer with them? And one question about the presentation, you showed a slide where the richness, about the richness and abundance of species and how they influence some ecosystem services. And there, I saw that the richness influenced more the, I believe, pollination, and the abundance influenced more the pest control. Do you have an hypothesis for why is that? And thank you.
So I've worked with pest control as well, but not within so much within agriculture systems. So for pollination, yes, I have a very clear hypothesis. So that there are several explanations why this diversity is important and why there is a synergistic effect between abundance and diversity, as I mentioned in a previous reply. So there is a lot of niche complementarity especially if we are talking about crops that are trees like mangoes, like coffee, and different bee species will forage on different parts. There are also interactions of behavior that enhance the effectiveness of the behavior. So that's very clear for me why there are positive effects of and interactive effects between richness and abundance. For biocontrol agents, what sometimes happens is that you have a dominant crop, and this dominant pest. So you need biocontrol agents to be very effective and they also need to be abundant, and the number of predators that is present that can feed on that. So usually pests are exotic, for example, parasitoids, they are much more specialized than parasitoids of insects. They are much more specialized than flower visitors. So flower visitors, some are specialized, but the ones that are in crops are all generalists. While the parasitoids of pests, they tend to be quite specialist, and there are not so many that can develop inside the body of the pest. If we are talking about parasitoids, and many of these studies that were integrated in this in the synthesis work were using parasitoids. So then it's normal that you need a lot of one species of parasitoid than several species. Yet, if there were more studies, and there is a lack of study, if there were more studies on the role of birds, on the role of spiders, on the role of bats as pest control. So these are three important pest control agents: spiders, the bats, and birds. And there is lack of studies on their effect on biocontrol. And if there were more study, because they tend to be more generalist, they are external predators, so they tend to be more generalist, then I would expect an effect similar to that was found in pollinators. So it has more to do with the nature, the specialization level of the consumers, pollinators versus parasitoids. I think that's the reason why we have these different patterns.
Thank you, Luisa. It was very clarifying. I asked that because I work with parasitoids, so I was thinking about exactly what you said. But I love that you mentioned the bats and birds. My advisor published a paper with another professor from UNB, saying just about the biocontrol of bats. Maybe you've heard of this work, and I would like to thank you a lot for your presentation and send greets from my advisors as well. They like your work very much. Raul Alman from Embrapa and Pedro Togni from UNB. And thanks a lot.
Thank you very much. And about your first question, as I said in the beginning, I'm open to questions also by email about the specific methods and analytical tools that I've used. So feel free to contact me and anybody else hearing this talk.
Luisa,
thank you.
Do you have more minutes? Because there are many questions here and one in Zoom, just to check about your schedule.
I'm okay for another 10-15 minutes.
Okay, great. So first I'll make the question that is here in Zoom. Then we will have Miguel, Rosa, and Tiffany. Was you? Yeah. So I will read the question that Gabriel Boldorini typed here. He said, thank you. And he liked the seminar, and he would like to ask you about if there are some functional traits, like syndromes, that can be used to better explain the variation of pollination than richness or abundance.
Sorry, can you repeat if there are better?
if there are other functional traits, for instance, syndromes, like I think pollination syndromes, that can be used to better explain the variation of pollination. I don't know what he means with variation of pollination, then richness or abundance. So Gabriel, if you want to join the question and make it clear, you can come in.
I think, well, if you want to complete, but I think I understood what he meant. So
Well,
yes.
yeah.
Okay, so Gabriel, yes, there are. For example, it would be very interesting to understand how functional diversity explains, if functional diversity explains better these patterns than richness. Because if we have 5 species, but they all act, for example, I told about this niche complementarity. So if we have a mango tree, if we have a coffee tree or coffee shrub, we have bees or flower visitors that will go to the top, others that will go to the bottom, others will be in more shaded areas, others more exposed. And this complementarity enhances, it's one of the main explanations why richness is so important. And obviously, I'm talking about complementarity that is associated with attributes, with traits. So if we had information, if we have 5 species that are all foraging on the same way, perhaps we wouldn't have these benefits. So yes, I think there is different traits that are related to effectiveness. Body size can be particularly important. So for example, if it is a fabaceae, the flower visitor needs to have a specific weight in order to open the kill, which is where the petals that protect the reproductive structure and effectively pollinates. If it is a Solanacea, it needs to buzz. And there are many other traits that relate with their foraging patterns. The problem that can be important. The main problem is that we really lack information on traits of pollinators in general, and this is a problem worldwide. It's not even just specific for the tropics or for Brazil, but there is lack of information, of complete information on traits, and we definitely need to get more of that.
Okay, I think it was, oh yeah, he says thanks. And then I think we have questions from here. Miguel, you can go.
Hi, Luisa. Thank you for the talk. It was a lot of information, but it was really good. I have a few questions, but I'm going to choose a single one, and the rest maybe I'll send you by email because they're more on the analytical part. I'm just really curious about in the first part of your talk, when you were talking about different processes of introduction and removal of species. And then you mentioned some sort of model selection, and I was curious about how it happened because it was a diagram with both paths about one of them decreasing the number of herbivores and the other one increasing the apparent competition and then decreasing the number of herbivores, if I'm not mistaken. And I was just curious about this model selection because I didn't understand if you chose between like these two very different models, as in because I interpreted that they were different hypotheses about the processes that were happening. And did you make a model selection to choose between them, or did you make multiple?
I did a model selection in that specific case to detect if the effect of the biocontrol agent was detectable over and above the effect of the invasive plant. So by comparing different models, you can understand which one explains more variability. I don't know how how, if you have used model selection before or not. But in model selection, we identify the model that is most parsimonious, which means that it explains most variability of data of our response variable, with the least number of terms in our model, with our least complex model, but also allows to understand when we have these variables that are correlated. They cannot be too correlated, but they are partially correlated, such as the case of the invasive plant and the biocontrol agent. There was obviously a correlation. You can use model selection to detect which variable is more likely to be explaining your response variable. And if you can detect the effect of one, so if the two are having very similar effects, they will annul each other, the effect in different directions, they will annul each other in this case, so they will become non-significant and they will not appear in the best models. When you have, in this case, we were able to detect that the model that only contained the biocontrol agent was more parsimonious, was explained much more variance than the model that contained both the biocontrol agent and the invasive plant, and even a model that only had the plant. So in this case, this model was even worse, that only had the plant. So by comparing the amount of variability that is explained by these different combinations, we are able to detect a best model, and therefore this is similar to then with pass analysis, we are able to quantify the amount of variability that is explained by each pass. But it's a first approach to detecting which is the most likely path of the effect.
Okay, thank you very much.
Hello, Luisa. Thanks for your talk. I was very amazed by the fact that you build the whole trajectory from local scale to global scale and past and present. And I was wondering, from linking your initial slides with the crop, first, the first work you presented was about invasive species and native species for conservation, the interaction between them and herbivores for conservation purposes. And then you went to crop yield and how pollination affects our crop benefits crop yields. And I was wondering that since most of our crop species are introduced species, how these crops affect the wild pollinators' biodiversity. You showed us that local biodiversity has positive effects on our crop yields, but I want to understand how our crop yields affect local biodiversity by changing the interactions between these wild species and some introduced pollinators that come with our crop species.
Okay, they change a lot. First, because of the deforestation that is caused, the replacement of natural habitat, obviously, and also because they tend to be monocultures. And so even the pollinators that use the crops, as well as the biocontrol agents that use the pests, they tend to be, but more with pollinators, they tend to be more generalized. So of the all amount of species that exists, for example, in Brazil, we have known at least 2,000 species of bees and many others, and only a small percentage will make use of these crops. Native, and I'm only talking about native pollinators. They will make use and they will use them as resource, but because it is monoculture, it offers these flower resources during a very short window. So usually this is not sufficient to maintain the population throughout the year. And this is why there are so accentuated declines of crop extension on pollinators. So many of the studies that, so I focused more on the effects of biodiversity on crop, but the first part of all these studies is to show the negative effects of crop expansion on pollinators or on biocontrol agents. And there is a very clear negative effect. So the more extensive crop fields are, the less pollinators we can find within the crop and also in the surrounding areas. So there are very strong negative effects. Also, very strong negative effects related with the insecticide use, with the fertilizer use. This is something that I'm quite interested lately. Usually most studies focus on insecticides, which are obviously quite damaging for pollinators, but also fertilizers can be quite damaging for pollinators and other biodiversity. And they are usually considered safe. So farmers, there is no regulation for fertilizers where there is some, not sufficient, but some for insecticides and other pesticides. For fertilizers, there isn't, and they are still also, they also have very important negative effects. So overall, yes, there are lots of negative impacts of farming on biodiversity as a whole, and of course, pollinators.
Thank you, Luisa.
And I think this is the last question. Go ahead, Tiffany.
Hi, Luisa. Thank you so much for the talk. I am more relieved that you comment on the differences between the scale of farmers, because I live in the state of Rondônia, which historically had as its beginning the incentive of the state for people to start to migrate there some decades ago, with the promise that they would have land as long as they cut down the forest and could maintain their crops and pasture. But these small farmers are not the real, how can I say, guilties for the majority of these problems that we were talking this afternoon. They are people who mostly make a living from the land for their subsistence. And many landowners, I mean, the big farmers, even practice this, I don't know how to say in English, but it's the grillaging to get more land. And another important thing to point today that I think is to mention the large enterprise such as hydroelectric dams, which have been growing in number in the Amazon region and are likely to increase in numbers in the near future. And because of this, we even have collections. Like you said, they are important. I agree with that. With a lot of material from the death of the large areas, and that also have changed the lives of artisanal fishermen forever too. So I would like to know what do you think we can do so that these social and economic disparities that are related to the use of the land can be more fair, to, I don't know, we can really protect our world.
Yeah, it's a big challenge. So certainly the type of farming that is causing more damage on ecosystem services in general, pollinators included, it's certainly this large, intensive type of farming. If we look at small scale, we could even say that the small scale farmers, when we evaluate the effect of pollinators on small scale farmers, sometimes we cannot detect it so easily because they don't have these very degraded areas. So the benefits are maintained in these small scale areas, while in the large scale areas they are really lost. But one of the things that so to try to answer more, so I think the two are very linked, and it's important to understand how the impacts of intensive farming are having not on biodiversity, yes, but also how they propagate to these small scale farmers, because then we talk about impacts on the social economic side. And if you look at distribution of funding across the different topics of interest of governmental focus, of course, biodiversity is the environment ministry. It's one of with the least funding allocated, and this is general in countries. It's not only from Brazil. And we need to understand that this loss of biodiversity is also having important social effects, and can also be considered in these other ministries, for example. One way that I think we can help to promote these local farmers, first is to recognize their importance, because, for example, family farming it's the main contributor for products. The area of plantation is much smaller than non-family farmers, but in terms of quantity of product, it makes the most contribution to national markets. So they are very important for maintaining the supermarkets and the markets in Brazil. And also it is possible to promote the products that are produced more sustainably, and these small scale farmers tend to be more sustainable, even if they don't want, simply because they don't have the money to invest on pesticides and fertilizers. So even just because of that, they end up being more sustainable. So if this type of practices is promoted, for example, with some type of recognition, subsidies as well, but also recognition that the consumer can see where the product is coming for, in order to give value to these more sustainable farmers, this might be a way to also increase the value and reduce a little bit the inequality. But it's a big challenge. It's a big challenge, and I wouldn't be able to answer in all the totality.
I think we had answered all the questions that raised hand here. There are more, and I would like to ask you, Luisa, if you can put your email address again in the slide for them to copy. Otherwise, I can send them if you... It's there. Okay, great. So, guys, if you have more questions, I think Luisa has other appointments. So, if you want to make more questions, you can write to her. I would like to thank you, Luisa, for your time and for accepting my invitation for the seminar. I had some questions as well. I can make them later. It was a very interesting seminar, and thank you so much for accepting coming here virtually and presenting to this group of students. Let's thank her.
Thank you, Flavia.
We are clapping right now, Luisa, but the talkers never listen to the claps.
Yes. No, but thank you, Flavia, and thank you all for the attention.
Thank you. Ciao, ciao.
Bye.